Alright, so skeletal muscle. Just wanted to make sure everybody knows what lecture we're on, because I'm going to be making a smooth muscle video here after this. So, skeletal muscle, I kind of drew out some stuff to save time because I'm a slow drawer. Skeletal muscle is kind of organized like this. So if I were to take a cross section of my bicep, this is what it would look like on a big level. You all know I like to go big to small in terms of picture size. So the first thing that we talked about is the coverings of collagen around each part of the muscle. So outside the entire muscle, outside the entire bicep for instance, would be the epimysium. So that's a covering of collagen. And then dividing like bundles of fibers or bundles of myofibrils is going to be the perimysium. Okay? And if we went one level deeper, we'd see in these bundles that the perimysium surrounds, there's individual cells. So myofibril is called, or is another word for a muscle cell. And if we went deeper and zoomed in, we'd see that it has a covering around each of those myofibrils. And those coverings are called the endomysium. So the endomysium surrounds an individual muscle or myofibril or muscle cell. And then inside or deeper to the endomysium, we have this thing called the basal lamina. The basal lamina is another layer that connects the outer part of the cells with the inner part, which is the sarcolemma. So it's called the sarcolemma. Alright, and if you take a closer look at this muscle fiber, which is just one muscle cell, you'd be able to see the sarcolemma. So that's like the plasma membrane. It's just a fancy way of saying the plasma membrane. And then around it, of course, would be your basal lamina. I just drew this same thing out down here so that I labeled it muscle fiber up there, but I wanted to draw in the myofibrils. So the myofibrils are the individual contractile portions, and there's going to be multiple of them in each muscle cell. Alright, and they are surrounded by the sarcolemma. So the basal lamina comes in play a little bit later here when we talk about the unit of myofibrils and then the smallest contractile unit of a muscle cell, which is the sarcomere. Okay, so the sarcomere is, if we pulled out one of these myofibrils and laid it longitudinally, so we're cutting down this way in a bicep, we'd see it like this. And that's where you see the striations. When we went to anatomy and histology, we saw like striations on skeletal muscle. That's where you get that. Okay? So, what does the basal lamina do for the cell? Well, it attaches to the sarcolemma via one specific protein called an integrin. So if you imagine, like this is our sarcolemma, which is the fancy word for the cell membrane, and this is our basal lamina, which is just like an outer covering. Like maybe think of it as like the outer cell wall on a gram-negative bacteria. There's going to be a little protein here called integrin that goes through the plasma membrane and hooks up the basal lamina to the rest of the cell, or the sarcolemma. And then we've also got another protein right next to it called the dystrophin that's going to attach each of the sarcomeres, we're going to see, to also the basal lamina and the cell membrane. Okay? So extra support, so that way when we're crunching up our muscle cell, we're keeping everything together. Because it's kind of like an accordion, you know, you got to be able to uncrunch itself when it's done. The basal lamina allows us to not fall apart as the cell kind of scrunches up and contracts. Alright, so the most important thing though in skeletal muscle physiology is how sliding filament theory works. So if we took just one sarcomere, which is the skeletal smallest portion of contraction in the skeletal muscle, and we zoomed in on it, it would look something like this. And again, not an artist. Okay, so on here, we call this the Z line, or Z line. It's called either one. Z lines are kind of the ends or the boundaries of the sarcomere. These are where our actin filaments attach. So actins are thin filaments, right? And they're not going to span the entire length of the sarcomere. They're just going to go out about halfway each. Okay, so that's our actin filament. Now, we've got another filament, as we may know, that's called the heavy filament. And that's starting in the center, and it spans the center of the sarcomere. And I'm shading it in a little bit harder so you guys can remember that it's the heavy chain in our sarcomere. Okay, so we got our myosin. And anchoring our myosin, since we're not connected directly to the Z line like, forgive me, actin is, we need something to attach us to it. Otherwise we're just floating out here in space and there's nothing attaching us anywhere. So we've got this elastic molecule, which I will draw in a different color, draw in red, called Titan or Tintin or whatever you want to call it. All I know is it starts with a T and it's not that important. It's elastic. So that is, I don't know how to spell it, Titan, Tintin, whatever you call it. Okay? And they just function to hold the heavy chains to the Z line. And they're going to be elastic. So they're not going to let this sarcomere drift away, drift apart. They're like a bungee cord. It can't get too far. It can stretch farther, like that's how your muscles stretch, but it cannot stretch to the point where this entire thing breaks apart. Okay? Alright, so we got that, we got that. Now we've got some different zones and lines. So we've got one here called the M line. The M line is the median or the middle of the sarcomere. Okay? It's just where everything's going to pull towards. So that's the M line. We've got the H zone, which I call the heavy zone. And that is just the place where the excuse me, kind of dry mouth today. The myosin filaments are, okay? And the myosin filaments are thick filaments, so they would weigh a lot, so that's how I remember that. The heavy zone is where the myosin filaments are because they are thick and heavy, okay? That's the H zone. Now, we have another area where they overlap. Okay, so the heavy zone was only including the portion where myosin was alone. Okay, so I kind of drew that a little bit too far. Now the portion of the sarcomere where there is actin filaments and there is myosin filaments is called the A zone. So where these two overlap, that's called the A zone. You'll usually see it labeled down here. Okay. And then for our last portion of the cell, we've got the only portion where myosin is not. So that is called the I band or I zone. So anything that doesn't have the myosin in it, including the rest of the sarcolemma, I zone. And that's a basic sarcomere, okay? So how does this cause a muscle to contract? Well, the way I always taught this in my human bio lab when I was teaching undergrads was always one specific way. I'd always remember rigor mortis. If you can remember rigor mortis, you understand the entire process of a neuromuscular junction functioning and it being activated, or the sarcomere, individual sarcomere, being contracted, okay? So this, the sarcomere, is going to be in a long chain of them, right? Just like this down here. And along some of these chains, or in between some of these chains, you're going to have things coming down called these T-tubules. They're just like invaginations into the cell that allow a depolarization to reach deeply into it. Because there's so many of these myofibrils inside of a single muscle fiber that we need to get deep into the cell to give the action potential to all of them, so they can all get the calcium they need to contract. Okay, I'm alluding a little bit here too far, but you get the point. So the T-tubule just dives deep down in the cell. We need to go back to the neuromuscular junction. So a neuromuscular junction is one motor neuron and the single muscle fiber it innervates, aka one muscle cell. Okay? So if we were to look at, let's say that this is a muscle fiber, so one muscle cell, and we had a nerve come in here, and that's my axon and synapse, we would see our motor end plate on the outside of the or muscle fiber right here. So right there would be the motor end plate. In the motor end plate is a bunch of receptors, and we went over those receptors. Those are nicotinic type 1 receptors for acetylcholine, which gets released from this synapse because this is the somatic nervous system, right? Acetylcholine gets and binds to those nicotinic M1 receptors and opens up the channel for sodium to rush into the cell. When sodium rushes into the cell, I'll put a little Na there, plus, goes in after a neurotransmitter has been released. It's going to rush down the T-tubule and rapidly depolarize this whole cell. Okay, so Na plus is going all the way down in through the cell, and we get the whole thing being depolarized, not just one little superficial portion of the muscle. Okay, because that would only contract one superficial part of it, and that's not beneficial to you. You want the whole thing to contract when you want it to. All right, so once that happens, what's our next step? Okay, so we got the T-tubule coming down. That's all good and well. Well, we need calcium. Okay, so when we get the T-tubule bringing down our signal, there's a thing next to it called the sarcoplasmic reticulum. The sarcoplasmic reticulum is just basically, it is the smooth endoplasmic reticulum. They just call it something funny in skeletal muscle. It contains a bunch of calcium in it. And it has little receptors on it that sense that the cell has become depolarized. When that happens, these calcium channels open up and allow our calcium to flood into the internal of the muscle fiber. Okay, so now we've got everything necessary to basically initiate a muscle contraction. We've got the calcium we need, and we know that muscle fibers have a lot of mitochondria, so we're definitely going to have a lot of ATP there. They're energy hogs. And now we can begin one of these cycles of sliding muscle filament theory. So if I zoomed on even more into the sarcomere, which you can do, you would see that each thick filament, which I am going to draw with the black marker here, has a bunch of these little heads on it. And then lining up right along them is our actin filament, or our thin filament. And this is our heavy, so our myosin. Now when our calcium comes in, if you zoom in on the actin filament, there's going to be something kind of really special there that allows, or is the reason for the calcium being needed for the cell. All right, so when that happens, we've got a binding protein, okay? I'm sorry, I got a text here. All right, so we've got this place for calcium to bind on this troponin, or actin thin filament. The place it binds is called troponin. Okay, so calcium is going to come in and bind the troponin. And I can't really draw it because, it's so small, but there's really good pictures out there of this on the internet if you want to look it up. But it's just a molecule of calcium binding to this one specific site, and they call it troponin. It's a protein. And what that does is it undergoes a conformational change and pulls, if we zoom in even more on the actin filament, you're going to see, let's say this is blown up a little bit, we're getting in even deeper, you're going to see these kind of other fibers wrapping around the thin filament site. So troponin, once it's bound by calcium, is going to pull this fiber called tropomyosin off of some very specific sites. So what's going to happen is now you're going to have a clear actin. It's going to have these little sites here for you to bind something. And these sites can be bound by the myosin heads, which are sitting right here. So when troponin pulls off tropomyosin, you've got these open binding sites. So that's a really good place for it to bind, and it's going to readily bind to those spots on actin, okay? So the reason I say that I start usually at rigor mortis is because what's going to allow a myosin head to bind and unbind from a actin? Well, there's one specific thing. So I always started it from here. I think of this as, I start kind of the opposite way, and I think of it as already bound, okay? They're going to be bound here. All right, so when you're bound here, or when you're unbound, excuse me, let me start here actually. When you're unbound, you're going to have a interaction. Oh shit, I should stop here.